All right, AP Biology. Let's talk about the operons. So first of all, you want to know what the general components of any operon are, okay? So of course you have genes that you want to express, right? These structural genes. There's 3 different genes that will transcribe and translate all at the same time. There's an on-off switch. You can call that the operator. So it's operating the process, whether it's on or off. Promoter sequence, like a TATA box, that's where the polymerase would bind. And then upstream is the regulation for this operon. Upstream, you'll have another promoter sequence for polymerase, which will then transcribe the gene for the repressor protein. The repressor protein binds the operator and turns it off. The operator is kind of like an on-off switch, and the repressor protein, the regulatory protein, regulates whether that switch is on or off, okay? So bacteria are tiny little things, and they need to deal with environmental changes very quickly, and putting a group of genes that are related to each other together makes it very efficient for them. That's the point of an operon, is efficiency. Not to make stuff when you don't need it, and to make stuff when you do need it. Very scientific stuff. Anyways, we're going to investigate this using the gene machine at Phet. Okay, so here we have some puzzle pieces that will fit into these different shapes. So remember, the operator is the on-off switch. I'm gonna start with my operator and see what happens. These 2 things floating around are my polymerase. So the polymerase will bind a promoter, right? So the promoter can bind the polymerase, and polymerase will go through and transcribe any genes that are associated with that section of the DNA. Okay, so here we are, we're making mRNA, right? The long strand is mRNA, which is then translated into a protein. This particular protein is lactase, let's call it. It breaks down lactose. When no lactose is present, this is kind of a waste of energy, right? Energy and materials. So let's regulate this so we don't make so much lactase when we don't need it. So we're going to add the regulatory protein in here. Here's the promoter sequence for the regulatory protein. This is the gene sequence for the regulatory protein. Polymerase binds, transcribes, creating the mRNA. The black strand is the mRNA, which is then translated into this repressor protein. So the repressor protein can bind at the operator site here. Then when polymerase binds this promoter sequence, it will not be able to transcribe the rest of the DNA. I'm not going to make any proteins that I don't necessarily need, right? So now I've regulated the amount of lactase in my environment. Over here, I'm going to add some lactose, and we'll see lactose can bind the regulatory protein. Notice how the regulatory protein changed its shape, right? This regulatory protein fits the operator. This one does not. So in the presence of lactose, the regulatory protein will release the operator, making it possible for polymerase to bind, and the polymerase then can transcribe the gene for the lactase. Polymerase can bind the gene for the lactase, creating lactase, which will then break down the lactose that is present in the area. Okay, so that's just one gene. So an operon is a set of genes that are all related. So let's go to this next one here. So let's go to this next one here. Again, we have our operator, our on-off switch. We have our promoter sequences where polymerase can bind to transcribe any genes that are present. So we'll transcribe and we'll make our regulatory protein. We can make our lactase protein, and we can make another protein that is associated with this process, which is a channel protein. So notice if you transcribe one, you transcribe them all. But not only do I make this lactase to break down the lactose, I also made these channel proteins which allow lactose to get through the membrane. Without the channel proteins, lactose would not be able to get through the membrane, and I wouldn't need the lactase to break it down in the first place, right? So an operon puts these genes that are related together. So when you transcribe one, you transcribe them all. It's all about efficiency. If there was a mutation, say, in this channel protein, you could imagine the lactose would not be able to enter the cell, and the operator would be, and the operon would be turned off all the time. Okay, so this is an inducible operon. We only turn it on when needed. Another example of an operon is a repressible operon, one that's turned off when you don't need it. So the trip operon is an example of that. So the trip operon is typically on. So it's constantly making tryptophan until we no longer need it. This red molecule here is my tryptophan. Here upstream, my regulatory sequence for my regulatory protein. So the trip repressor protein. Notice this shape will not fit the operator. This shape will not fit the operator. So the operator would typically not, the repressor protein would not be sitting on the operator. The operator would be open and we would transcribe all we want. When we've made enough of our tryptophan will bind to our repressor protein, bind allosterically opposite the substrate binding here. Notice it changes its shape and now fits the operator. So now this shape, the repressor protein, will turn the operator off and will no longer transcribe tryptophan because we have plenty. We don't need any more. Okay, so we call tryptophan in this case a co-repressor. The repressor protein doesn't fit by itself, but once you add the co-repressor, the repressor fits the operator. Okay, so you have the operon that's inducible when something is present that needs to be broken down, and you have the operon that is repressible, usually a synthesis reaction. When you've made enough, you don't need to make any more. So those are our two model operons. I hope that was helpful.